Last time, we introduced you to the revolutionary lab-grown brain chip from FinalSpark, a technology that blurred the lines between biology and AI. But today, we're diving even deeper. In this episode, we'll show you how these living brain cells are now controlling a virtual butterfly in real time. It's a breakthrough that's not just redefining computing, but raising profound questions about the future of intelligence itself. Let's explore what's next in this incredible journey, as thrilling as it is unsettling. But before we delve into the virtual butterfly, we need to understand the key component driving this innovation: brain organoids. These are tiny pea-sized creatures grown in laboratories using human stem cells. While they're not complete brains, these organoids contain approximately 10,000 neurons that can form networks and respond to stimuli. Think of them like simplified versions of brain tissue that scientists can use to study and experiment with. What makes this grand achievement possible is FinalSpark's innovative neuro platform, which they've dubbed the world's first wetware cloud platform. This system uses 16 brain organoids, each carefully maintained in specialized incubators at 37 degrees Celsius or 98.6 Fahrenheit. The platform creates a two-way communication channel between these living cells and computer systems through something called a multi-electrode array, or MEA. With this revolutionary platform in place, let's dive deeper into how exactly these brain organoids are controlling our virtual butterfly, creating an unprecedented bridge between biological thought and digital action. So, how does the virtual butterfly work? At its core, the system features a meticulously designed 3D virtual world, where a butterfly exists as a dynamic digital entity. This isn't just any computer simulation. It's a sophisticated environment that responds to both human input and biological processing in real time. The interaction begins when a human participant clicks within the virtual environment. The system's software immediately performs complex calculations to determine whether this click falls within the butterfly's designated field of view, similar to how a real butterfly would perceive movement in its environment. This calculation takes into account the butterfly's current position, orientation, and the natural limitations of its simulated visual field, creating a realistic representation of biological constraints within the digital space. Once the system confirms the click's relevance, this information undergoes a fascinating transformation. The digital signal is converted into electrical impulses that can be understood by the brain organoids through the sophisticated MEA system. These electrical signals mirror the kind of neural stimulation that occurs in biological systems, allowing the lab-grown neurons to process the information in a way that's natural to them. The brain organoids then process this input through their neural networks, creating patterns of electrical activity that represent their response to the stimulation. This response isn't pre-programmed or deterministic. It's a genuine biological computation performed by living neurons. The system then interprets these neural responses and translates them back into digital commands that control the butterfly's movement. If the neurons respond positively to the stimulation, the butterfly gracefully navigates towards the clicked location. If not, it continues its journey with randomized movement patterns that maintain the simulation's natural feel. What makes this system truly revolutionary is the genuine biological decision-making at its heart. While the butterfly's movements themselves are executed through programmed animations, the crucial decisions about where and when to move come directly from the biological neural network. This creates a unique hybrid system, where artificial and biological intelligence work in harmony, each handling the aspects that they're best suited for. While the butterfly's movement is fascinating to observe, the true marvel lies in the underlying biological neural networks that make this possible. Let's explore why these networks represent such a groundbreaking advance in computing technology. The most striking benefit lies in their incredible energy efficiency. While the human brain, nature's most sophisticated computing system, operates on a mere 20 watts of power, about the same as a dim light bulb, modern supercomputers like the HPE Frontier require up to 21 megawatts, equivalent to the power consumption of a small city. This astronomical difference in energy consumption highlights the potential for BNNs to revolutionize sustainable computing. The adaptability of biological neural networks sets them apart in remarkable ways. Unlike traditional computers that excel at specific programmed tasks but struggle with variation, BNNs demonstrate an extraordinary ability to recognize patterns and adapt to new situations. This adaptability stems from their biological nature, which enables them to process information in ways that mirror natural learning processes. They can identify subtle patterns across diverse datasets and apply learned principles to entirely new situations, a capability that current artificial neural networks still struggle to achieve consistently. Perhaps most impressive is their capacity for zero-shot learning, the ability to handle entirely new scenarios without prior training. This is something that traditional AI systems find extremely challenging, often requiring extensive training data for even minor adaptations. BNNs, however, can naturally generalize from existing knowledge to new situations, much like how humans can understand new concepts based on related previous experiences. The self-repair capabilities and neuroplasticity of biological networks represent another groundbreaking advantage. Unlike traditional computer hardware that degrades over time and requires replacement when damaged, biological networks can actively adapt, repair, and reorganize themselves. This neuroplasticity allows them to maintain functionality even when parts of the network are compromised, and in many cases, they can optimize their structure for better performance over time. These impressive capabilities of biological neural networks are just part of the story. To fully appreciate this breakthrough, we need to examine the sophisticated technical infrastructure that makes this biological digital interface possible. At its foundation lies an array of 4 multi-electrode arrays, each carefully designed to house 4 brain organoids. Each organoid interfaces with 8 precisely positioned electrodes, creating a complex network of 128 communication points that enable seamless two-way interaction between the biological and digital components of the system. The digital-analog conversion system forms a crucial bridge between these two worlds, sampling biological signals at an impressive rate of 30,000 times per second. This high-frequency sampling ensures that even the subtlest neural responses are captured and translated accurately into digital information. The precision of this system is further enhanced by 16-bit resolution, allowing for extremely detailed measurement of the organoids' electrical activity. Supporting these core components is a sophisticated microfluidic life support system that maintains optimal conditions for the brain organoids. This system carefully regulates temperature, pH levels, and nutrient delivery, ensuring the biological components remain healthy and responsive. The entire setup is monitored by high-resolution cameras that allow researchers to visually inspect the organoids in real time, providing crucial feedback about their physical condition and development. Tying all of these elements together is a comprehensive software stack that orchestrates the entire system. This software handles everything from processing raw neural signals to managing the virtual environment and coordinating the various subsystems. It includes sophisticated algorithms for signal processing, pattern recognition, and real-time decision-making, all working in concert to create a seamless interface between the biological and digital domains. The software also includes detailed monitoring and calibration tools that ensure all components of the system operate within optimal parameters. While this technical achievement is remarkable, what makes it truly revolutionary is how FinalSpark has chosen to share it with the scientific community. They're offering access to their Neuro platform to researchers and institutions for $500 per month per user. This accessibility could accelerate development in the field by allowing more researchers to experiment with and build upon the technology. As we look ahead, this breakthrough represents more than just a novel way to control a virtual butterfly. It's a stepping stone towards a future where biological and digital computing might work together in harmony. And there you have it! Living brain cells controlling a virtual butterfly. A glimpse into a future we're just beginning to understand. But this is only the start. Imagine what this technology could mean for AI, human augmentation, or even redefining life itself. The question isn't just how far can we go, it's whether we're ready for what comes next. But what do you think? Are we unlocking the future or opening a door that we can't close? Share your thoughts in the comments down below. And if you're as fascinated or as unsettled as we are, hit that subscribe button and turn on notifications. The next big breakthrough might be closer than you think, and you won't want to miss it. See you next time!